At Adiseo, we cherish the work we carry out with animals. And thanks to us, chickens and pigs grow with less feed and in a healthier way. Cows produce milk in greater quantity and quality for a longer period. And fish and shrimp now have fewer diseases and grow faster. These animals contribute to feed 8 billion people and have enabled us to become a global leader in the production of nutritional solutions for animal feed. One of the leaders in methionine, an essential amino acid and protected methionine for ruminants. One of the key players in the vitamins and specialty products designed to foster animal health and welfare. Our story began in France with Alimentation équilibrée in Commentry in 1939. Then in 1971, the company is acquired by Rhône-Poulenc, becoming its animal feed division. In 2002, acquired by an investment fund and renamed Adiseo, the company regains its independence. And in 2006, we joined the Blue Star Group, thus asserting our position in Asia and China especially. Adiseo is a unique company in many aspects. Our roots are in France, but our products come from Europe and China, and 95% of them are exported to over 110 countries and have more than 3,900 customers worldwide. We are a medium-sized company with around 1.5 billion euros turnover and almost 2,200 people working for Adiseo. Our uniqueness also comes from our investments in both industry and research, guaranteeing a competitive and innovative product and service offering. 1.7 billion euros have been invested since 2007. Our success is also due to our dedication to essential values: team spirit, creativity, integrity, commitment, and results. Over the years, our company has changed, developed, and grown, putting safety and sustainability as top priorities. Safety of our employees, safety of our plants and of our research centres, safety of our products. We never do any compromise with safety. Aware of present and future economic, environmental and social issues, Adiseo has developed a growth strategy that is clearly in line with the UN's Sustainable Development Goals. Building on its strengths and successes, Adiseo strives to be a partner of choice in animal nutrition, with women and men who make the difference every day to feed the planet in a high-quality, affordable, safe and sustainable way.
Hello, this is Dr. Brian Sloan, today's moderator. I am the Global Director of Adiseo's ruminant amino acid business. On behalf of Adiseo, I would just want to extend my thanks to the DCRC for the invitation to participate in one of their key educational webinars. Today, we will share with you information and the scientific background concerning the roles and importance of dietary methionine and selenomethionine on health and reproduction. We have two invited well-renowned experts to talk on each of these topics. Without further ado, I would like to introduce our first speaker. Dr. Phil Cardoso is an associate professor at the University of Illinois, Champaign-Urbana, Illinois. He received his DVM and master's from the Universidade Federal do Rio Grande do Sul in Brazil, and his PhD from the University of Illinois. Since 2012, Phil has established a unique program that seamlessly blends his teaching extension and research efforts. The impact of Phil's dairy science program is in placing students in applied positions and academia. Phil and his students have published over 75 peer-reviewed manuscripts and 3 invited book chapters to date. The program builds from questions asked by dairy producers and focuses on having the dairy cow's diet as a medical prescription for performance, health, and reproduction. This is achieved by understanding the impact of nutritional metabolism, reproduction, and health in dairy cows, as well as the mechanisms of metabolic adaptation to stressors and forage quality. Without further ado, Phil, the floor is yours.
Thank you very much, Brian. Very good to see you. Hopefully soon we're going to all be doing those in person. And let's go ahead and share my presentation. So I have this first 30 minutes with you guys. We're going to go start, share some, optimize. I want to do this. Can you give me a hint if you guys are seeing the whole screen?
Looks
We
great.
see it, Phil. We see it, Phil.
Okay, I even have hair, so it's even better. Better than normal. Better than everything we can get. Okay, so here we go. So I'm not, by the title, please don't think that selenomethionine is less important than methionine. It's just to highlight my session of this presentation, the first 30 minutes, right? So I have this task of trying to connect nutrition and reproduction, and we are trying to do that for a long time, but I actually found a video that can exemplify and remind me back my years from practicing veterinarian back in Brazil, working with nutrition and reproduction. So I want to play this video for you guys. Hopefully you can see it.
But I tried everything just to get along with you. But I'm gonna do just what I'm gonna do.
So that's pretty much what is saying there. That wording was in Portuguese. Actually, there was a commercial for Fiat back in Brazil. And guess what? Now I own a Ram truck and Fiat owns my Ram at this moment. So big company getting even bigger. But that's some of the things that connects, and that's your passion, right? So here we go, passion, nutrition, and reproduction. One thing that I want to call attention, because we're going to be talking about amino acids, and there's always this question about what are the amino acids that are limiting, which ones are going to be the next one. So what I'm pointing out here is that depending on the output, right, that we have for those species, that limiting nutrient is going to be different. And here I'm pointing out amino acids. However, we could think about trace minerals. Probably having wool as an output is going to be very different than having meat on those trace minerals. And amino acid is the same thing, right? So if I think about, for example, papers talking about how important arginine is to reproduction and to the offspring, that is going to be very well related to arginine in for wool and for lambs. However, cows, since milk is the main output, it's not saying that arginine doesn't have a whole or other amino acids. It's just saying that, hey, the output, at least since 2001, where the last NRC came out, they already told us that methionine and lysine are the most limiting amino acids. So that's why we need to kind of focus and direct our attention to those two amino acids. And we are going to come back to those in a few. Another point is to consider is the timing. So here you're seeing an example of arginine of fall lambing ewes, right? That may mean that the spring, the ones that fall in the spring, that may be a little bit, the lamb in the spring may be a little bit different. And with cows also, depending on which stage of lactation or that production cycle, that may be a little bit different. But we do know that the transition period, 3 weeks before, 3 weeks after, is very challenging for cows. But some cows are successful in that transition, and they can achieve, and we can get more in depth into that, but they can achieve a high milk yield and successful reproduction as well. However, we through nutrition can alleviate that negative energy balance that we know very well, and also that negative protein balance that is happening at the same time those first 4 weeks after calving. But we need to recognize that reproduction is not going to be at the top of the scale or the top of the pyramid in importance for that animal. Actually, it's going to be one of the last. So we do need to pay attention and configure or strategize a diet that suffices all those nutrients, right? Specific related to that output for that species. In this case, we're going to talk about dairy cattle. Now, one thing that is very important, I think, this council has been very involved in a lot of the research out there, especially on synchronization protocols and how to get the cow pregnant. And I think with double ovasync, we really moved to get cows pregnant. And I think we still have that question, okay, the cow is pregnant, how can we keep her pregnant now? And we are seeing a lot of this embryonic loss. And a very nice paper by Santos, a while ago already, he put together some of his trials. And the good thing about this table is that he was checking, and his students' pregnancy around 30 days after AI, and also around 45 days. So here is the difference between first check and second recheck. And one thing I want to highlight is that anywhere from 10 to 20% was typical to see this loss of this embryonic loss. So that's already one question that I throw out there, is that you as a consultant, veterinarian, farmer, nutritionist, you need to know your number. If you're doing 30 and 60 days, 40 days and 60, I don't know, but something in between there that you can have an assessment on that. And of course, there are multifactorial event that is embryonic loss, but we can probably help that with nutrition. And I'm going to show you today one example of that. Of course, there are several diseases that will require other fixes, but nutrition can play a role. So you make sure to cover your bases there. And as you can imagine, it has some cost associated with it. That's why farmers are very keen into reproduction, milk yield, and also cows being healthy, because they know profit comes out of performing well on those three areas, right? And De Vries out of Albert, out of Florida, determined that on that first month, you can be losing around $150 to $360 per pregnancy loss. As time goes by, that gets more expensive. And of course, the variation in depending on feed costs and replacement. So there is some variation there, but there is a cost associated with losing that. So it's not a good deal, right? The other thing is, if we are trying to suffice, or we are trying to provide to the cow enough amino acid, and we may say that's through protein, feeding a lot of protein that goes back to high milk urea nitrogen, it is shown to be negatively associated with that first breeding. So if that first breeding is around 70 to 80 days, that can be compromising cows with a high MUN or a high nitrogen circulating, or feeding a high protein diet, right? They didn't find any effect on the subsequent AIs. So the second or third, they didn't find an association. And they did in quite a bit of cows. So they did over 10,000 cows on that data set. And one good thing that they did here was to separate those cows in high producing cows, moderate and low producing. They also did separate them in seasons, the spring or fall. And just as an example here, a high producing cow, over 100 pounds of milk per day, and if she is at 20 milligrams per deciliter on MUNs, that means that she will have 35% conception rate. That's on the average on this data set. That talking about in the spring, it's going to be a little bit lower in the fall, but I don't want to get into the season effect, but I want to get into the fact that if that cow was producing 56 pounds of milk per day, now the same 20 milligrams per deciliter concentration of nitrogen in that milk, that would lead the cow to have a 40% conception rate. So that brings the idea that we need to keep in mind that milligrams per deciliter is how much nitrogen you have in that milk. As you have more milk, you're going to have more nitrogen circulating in the tissues of the cow, including the liver, including the uterus. So that's why the effect is going to be much larger for cows that produce a lot of milk. So farms perhaps that you are on pasture or milk production due to limited genetics are lower, you may be a little bit more forgiving on that MUN if you're tracking that. But of course, 20 is just an example here. That's not. You guys probably, where is my little laser? Here it is. You guys probably are going to be around 10 to 14. That's where you're going to be targeting that in your farm. But the association is linear. So if the cows are producing less, you can be a little bit more forgiving with that MUN. But in a broader sense, feeding a lot of protein is not a good idea, right? And why is that effect on reproduction? There's this very nice paper here by Rhodes where he got some heifers. And one thing that he did was some of them, he was injecting or infusing them with saline. So pretty much a control, right? A placebo. And he was following the PUN or plasmourea nitrogen that correlates really well with milk urea nitrogen, circulating nitrogen in the cow. And as you can see, it just kept consistent as he did the infusion, and the pH in the uterus he was always measuring, and that kept consistent. Now, the other set of animals, what they did was to infuse urea as a source of nitrogen to simulate cows eating a lot of nitrogen, right? Consuming a lot of nitrogen. And then you can see that as designed, plasmourea nitrogen goes up with time and the uterine pH goes down, right? So as you're changing this, having that change in osmolality in the tissues, because of that nitrogen now needs to be metabolized, you're going to have that exchange of hydrogens, and you're going to have the water pretty much being changed to make sure that nitrogen is diluted in the tissues. And that's where you see this pH. And then there's some other research on even in vitro data saying that that change in pH is not a good deal for conception rates, right? So one of the reasons perhaps that high milk urea nitrogen, high PUN affects pregnancies is because of this change in uterine pH. And when we talk about pregnancies, Walsh identified at least 7 factors that are important, right, to get that cow pregnant. So we understand very well body condition score and how does that affect the cow's health and reproduction, and we understand the nutrition connection there. But today, what I would like to talk about is more about this uterine environment, not necessarily prostaglandin, but uterine environment. And this idea that a large embryo could have a better production of this interferon tau that's going to signal the cow about the pregnancy, right? So if you think about that cow is bred, now that conceptus is going to be traveling, is going to be reaching out and touching that uterine wall. There needs to be a signal around that 14 to 18 days telling the cow not to cycle again, assuming she's doing that every 21, 23 days. There needs to be something saying her, hey, don't come in heat again. And the signal is this interferon tau. And there are associations between that and the size of the embryo. Larger embryos, more signaling to the cow. So we are going to go back to this in the end of this presentation. So this is one thing that we can impact with nutrition. So what is the right diet to feed those cows, especially those dry cows, to impact positively the transition period, right? I go over in a symposium review with other co-authors. So you guys, if you want more details about it, you can read there in Journal of Dairy Science. But also, and the DCRC is involved with that book, Dairy Cattle Fertility from Horst Darren. I have a chapter there on nutrition and fertility, and we talk a little bit about in more depth what I'm presenting here today. So feel free to go out there and reach to those publications. But crude protein, we talk about 12 to 14% of the dry matter. And remember, crude protein is nitrogen multiplied by 625 in your diet. And here is a good point or a good data from Dr. Clark and Ignacio in 2005, talking about why it's not a good idea. So 2 information that I get from this graph here. So this is the dietary crude protein. and how much milk those cows are producing, right? Data from several trials. So if we think about 18% crude protein, I can have cows at 23 kilos per day, so roughly 46 pounds of milk per day, but I can find cows at roughly 80 pounds. So I go from 46 to 80 pounds at the same crude protein. And if I just invert this conception, then I can have cows at 35 kilos or roughly 70 pounds that are eating 13% crude protein, or they are eating pretty much 26% crude protein. So the point is, crude protein is not a good predictor of milk yield or performance, right? So that means that I'm not even talking about what is the RUP on those diets, what is the RDP. So crude protein is not good to prevent. And if you wonder why we always talk about 18, 20% crude protein for dairy cows, is because that's where that curve starts flattening, right? So they said, yeah, let's shoot there because that's a good point. But look the variation in this data. So we need to describe better our feeds, our diets. We cannot talk about crude protein anymore, right? So the suggestion is just what I talked to you. Metabolizable protein is going to give you a better idea on how much microbial protein plus everything bypassing the rumen and some amino acids getting to the duodenum of the cow. So I want to know how much that pre-fresh cow is absorbing in grams. And this is going to give me an idea of a good profile of that protein. Remember, proteins are made of amino acids, but I need to make sure that the two main limiting amino acids in that protein that is going to depend on which sources I use in the diet are going to be fulfilled, right? And the whole 1,200 grams can give me, at this point for us, an idea that the other amino acids are taken care of. If I don't feed the other ones and just feed methionine and lysine, that usually is not going to go well as well. We can talk more about it if we have time in the end. Now, going to the health, there is the group from Italy, Trevisi and Bertoni in the past. What they are developing was, hey, if I collect blood samples before calving and 2 times after calving, and I have albumin, cholesterol, and bilirubin, I can get to this liver functionality index, or LFI. And they identify that, hey, cows with low LFI, they are the ones having trouble. That means that the liver is not working properly. And the cows with the high LFI, they are having a smooth transition. That means that the liver is not overwhelmed. And in this research by Zizi, what he found was that a tendency that cows that were receiving methionine, rumen protected methionine, they were more in the high LFI group. So let me share with you some data about it, okay? One thing they found, so in white here, you see the high LFI. Those cows with a better liver functionality, they eat more before calving, they eat more after calving, and they produce more milk after calving. An indication now of metabolism, like I mentioned before, they have a higher concentration of methionine circulating, the cows that are performing better with the liver, and they have lower 3-methylhistidine that is an indicator of muscle depletion. So that means that throughout that transition period from -10 to 28, they are using less body reserves from muscle compared to cows in the low LFI. And some idea on the inflammation is that those cows in the high LFI, they have a lower acute phase protein, like haptoglobin in this example, and they have higher pro-inflammatory molecules like pyroxenase. Is just pretty much, hey, if something happens here, we are ready to attack. But that doesn't mean that we are out of sense, right? So that's kind of idea of the good inflammation, bad inflammation. That's what we are playing here with pyroxenase. So very interesting. I wish we would have like a cow side test that could tell you if a cow just calved, what is the chances of she being in one way or another. But I think we're going to see much more coming from this to talk about the health of the cow. This can connect to well-being, so welfare. So I think we're going to be seeing more of this type of research here. Very interesting. One thing that we've done with cows that were fed rumen-protected methionine is this discovery of this neutrophil extracellular traps, or NET by short. So here we did some histology with those cows, right? So it seems like these control cows, they are having a little bit more activity on day 30, right? So you can see the number of cells per gland in the uterus, the perimeter, the epithelial heights are a little bit harder to understand here since we didn't measure all the hormones, but it seems like for some reason that tissue was more metabolically active. But one thing to be concerned is that since that control cows at the 73 days in milk, so when you're getting them ready to be bred, they had more of those PMNs, or those neutrophils, polymorphonuclears, or a tendency to have higher. And that's probably not something we would like to due to interference with that conception, right? And this is something just to be aware of that I think we're going to be seeing more in the future, is that we were able to identify here this NETs, right? So that can be activated for several factors. That was not associated with methionine or not, so we just saw it happening in the uterus. Then you can see these little green things here called, that's the elastase, that enzyme that realizes this trap. So imagine a spider web trying to trap all these pathogens, like a bacteria, for example. And we don't know if that's good or bad, but we know that that's happening in the uterus now. We knew before that that would happen in the circulation of the cow. But so if this is happening here, and if I get another information and give you from Matt Luce's lab, where they identify that cows that were repeat breeders, or cows that were having problems breeding, they had a higher number of this lymphocytic foci that they call. So all this mass that you're seeing here are this accumulation. So they saw that, hey, a small or intermediate foci in the histologic had something related to embryonic loss. So that could be something that starts as a NET. So those neutrophils are extrapolating on their defenses, and now you have all this foci, and now you have parts of the uterine tissue that is not secreting anything, and then you may have these problems with placenta development and embryonic death. So something to watch in the future, but at this point, we couldn't find any association with methionine, even though we didn't have a big number of cows, a large number of cows, right? The other thing then we did in a larger pool is to, again, use that pap smear, that brush, collect some cells from the body of the uterus and measure those PMNs and count them as a gold standard for endometritis. And we saw that at 15 days in milk, cows that were receiving methionine, they had higher PMNs, but that changed throughout the lactation. So another concept that is important to know when in the life cycle of the cow, which days in milk she is, because at 15 days in milk, higher PMN with, of course, no fever, no loss of intake of milk yield. That means that the cow is perhaps the mounting process of inflammatory process to do the uterine regeneration, for example. At 73 days in milk, that's not something you want to have happening because of all this issue with conception. 15 days in milk, you don't gonna have any worries about trying to breed that cow, right? Just to make sure that cow gets recovered as soon as possible. And in 2014, a conference in Ireland, actually, Dr. LeBlanc from Guelph, he suggested that the healthy cows, when I'm talking about the uterine inflammation, are the ones that can mount a response, but then they quit that response because they clear the inflammation. Now, the problem cows are the ones that keep responding, and the cows that don't even respond at all because they perhaps have not those enough of those nutrients to build a response, right? And more recently, we just had this paper accepted that where we collected and we did some gene expression of the cytology of those cells that we collect in the brush, but also we did a uterine biopsy, and we want to see how are those related. So I'm not going to get too much in details here, but you can see that, yes, you have higher expression of that methionine receptor in that cell. There is some proliferation going on higher in RPM, less inflammation, happening in those cells in the RPM or methionine group compared to control. And in the endometrium, you have some similarities, or yes, you still have more proliferation on that uterine tissue. You have more metabolism going on this homocysteine. You have perhaps better capacity for nutrient like glucose, conversion of cholesterol, and also a polypoprotein related to fat transport and less inflammation. So very interesting that that amino acid that we know is required can have such an effect on the uterine tissue as well compared to not being fed rumen-protected methionine. Those cows at 73 days in milk, we actually harvested, we superovulated them, collected the embryos from them, and summarizing here, we found that the embryos from cows on methionine, they had more lipid than control. It's not saying that the embryo is fat, it's just saying it had more lipids, right? Then trying to understand why more lipids is something that we are doing it at this moment. One thing we did was to use this MALDI technique in partnership with Purdue University, where you can put each embryo, shoot a laser on it and see how it reflects. And then you have this detector that it can map what is the fatty acid profile of that embryo. So what we get back is just this heat map that is going to identify what type of fatty acids we are having on those individual embryos. So I don't have to pull the embryos, I can do one by one. And this is what we have. So not totally sure yet what does this mean, but definitely I think I'm going more on the side of that embryo have a type of fatty acid that can communicate or be involved more in signaling rather than accumulating energy for the future, right? So there is this shift in having higher polyunsaturated fatty acids inside that embryo. And we also had the biopsies from the uterine tissue. Now, with more tissue, we can go more in depth a little bit. So you can see that at 15 days in milk, there's no difference if the cows got methionine or not on the fatty acid profile. But as we go on the lactation overall, we are having more saturated or unsaturated fatty acids in that tissue. So methionine being able to perhaps be part of that apolipoprotein. Remember that fat to be transported in the body, it's extremely insoluble. So we need to be attached to that protein. So perhaps that methionine is needed, it's required for that protein, apolipoprotein, to form and now be able to transport that fat around and be able to get to the uterine tissue, perhaps now to nourish and to play a role in the nutrition of that embryo and later on fetus. Very interesting connecting different worlds, right? So we go north of here, Illinois, where I am. We go to Wisconsin. Milo Wiltbank's group, in collaboration with Dr. Shaver, they harvested embryos from cows, fed methionine or not, after calving. And what they found, they did this transcriptomics and say, hey, tell me which genes are up, which genes are down. Tell me everything. So they found 30 that were different. And guess what? With methionine, 2 were altered. 1, we don't know what it is yet. You know, you just go fishing what's going on there. But the second one that was up was a polypoprotein tree-like. So they also identified some association with lipids. And in this case, the embryos from those cows receiving methionine had higher expression of polypoprotein tree-like. And just reminding you, this is the whole complex that we need to be transporting fat. So imagine a cow during a transition period, how much fat needs to be transported. Not having methionine can be a big deal on the liver and also other tissues like the uterus, right? And coming more here to the end of my talk, you can see that we also saw some of the effects of methionine on the placental tissue. So just remember that in utero nutrition of the fetus is happening through the placenta. So there are receptors not only for amino acids, but for glucose, fatty acids, vitamins. And we then harvest the placenta of those cows. As soon as they calved, we collect the placenta, did some work on protein abundance. And what we were able to see is that the cows that received methionine they had a higher expression or realization of that phosphorylated mTOR over mTOR. So that means that protein synthesis was more activated in the placenta of the cows receiving methionine during the dry period. And not a coincidence, but also reflected on those calves when born from the methionine cows, a little difference, around 2 to 3 kilos there, but a difference, right? Playing a role in this nutrition of the calf within the uterus is still. No differences in this, dystocia, anything like that, but a difference that we were able to capture statistically versus a tendency here in the protein synthesis. And my last one here, coming back to the state north of us from the Dr. Wiltbanks and Shaver work, is that they fed cows from 30 days after calving to 128 days, feeding methionine or not. And I'm bringing this paper here because it's very important. They were able to do that in a commercial farm, right? So a lot of work. You have to make sure the cows are eating the top dress with the amino acids. So a lot of attention needs to go here. And what they found, of course, a lot of cows, 300 cows, they were able to recruit primiparous and multiparous cows. But the main thing that I'm going to bring to you is that when they palpate those cows or when they check for pregnancy at 28 days and later on at 61, primiparous cows, no difference. But look the difference here in multiparous. So it went from 19% embryonic loss to 6% if cows were fed rumen-protected methionine. And here, mainly, remember that I told you in the beginning, this is typical. It's not off the charts. 10 to 20%, we said, that's pretty typical. So we need to know, and it seems like methionine can impact that. But the good thing here is that they went a little bit further. And actually, when they were checking those pregnancies, right, they were able to measure the volume of that embryo. And guess what? Primiparous, no difference in the size and the volume of that vesicule in that embryo. However, multiparous cows, the ones that were receiving rumen-protected methionine, the embryo was larger than the control. So probably meaning that signaling of pregnancy was more present in here. And there is research that I didn't show today that shows how important methionine is for the embryonic growth. So all the media that you guys use for embryo transfer, IVF, I will bet you're gonna have to fight not to find methionine in those media, right? But there's another area of opportunity on manipulate that media to impact that embryo survival when you are transferring an embryo or doing that fertilization, but different story there. So take home message here. I think it's very important for us to focus on giving the farmers what they need. As a veterinarian or a nutrition, they're really going to focus on those 3 things. I think making sure that the cow is eating the overall diet is important to prevent any of those problems. And here is a little bit on how we see that. So we think that 1,200 grams at least of metabolizable protein, you're going to need a software for that, a model, right, to tell you how much is that. We here are using CNCPS, associating that grams of amino acid with energy. Where is that energy coming from? We're going to be telling more in the future if it is from carbohydrate, sugar, fats. For now, it's energy, right? And here a little bit of the reasoning of the best, where these numbers are coming from. And it's pretty much is going to be your best efficiency of utilization, or how much you feed and how much the cow can use. Even though sometimes you don't want to be efficient, right? You want to focus on performance. So you may play with that number. There is some variation there that you may want to play, but you have to start somewhere. And here's my suggestion for you. And with time, you're going to be learning and you're going to be getting your own perspectives on some farms, maybe a better number, a little bit higher, some farms a little bit lower, right? But we need to start somewhere. And we are plugging this from lactating cows into dry cows. And that's how we are doing right now. So a lot of the papers you saw here, that's the concept we are using. So with that, I would like to thank the DCRC, thank Ediseo, and I'd be more than happy to chat about it now or in the end, whenever you guys want to pull me in.
Thanks, Phil, for a great presentation. I'm sure this has provoked some very good questions from the audience. If you have questions for Phil, please feel free to enter them in the Q&A. We have a few came in, but we're going to set the bar at 10 so that nobody gets any credits unless we get 10 questions at the end. Maybe that will motivate some of you to ask some more questions. But without further ado, I'd like to introduce the second speaker today. Dr. Darren Juniper worked within the University of Reading's Animal Science Research Division. He gained his PhD from Reading in 2003, entitled Diet and Endocrine Responses in Beef Cattle. He has been involved in animal production research for over 30 years, and in this time has worked with most classes of domestic farmed livestock, both ruminant and monogastric, covering a diverse range of research topics. But more recently, over the past 15 years, he has worked on the area of trace element nutrition, primarily selenium and iodine in animal diets. His selenium-based research has focused on the effects of selenium source on aspects of animal and human health, as well as the deposition of selenium in the products and post-mortem tissues of both ruminant and monogastric livestock. Darren, the floor is yours.
Thank you very much, Brian. And thank you to Phil for an interesting introduction. I'm not sure how well I'll actually live up to his introductory video. I don't have anything nearly as flash as that. So what I thought I would start with is a brief introduction as to what selenium is, and then lead on into maybe the benefits of selenomethionine, and certainly in terms of the effects that actually has on health and fertility of dairy cattle. So very simply, first question sometimes I ask students when asking about nutrition is, what is selenium? Many of them have never heard of it, none of them, well, except if they've watched the film Evolution. If you go to Wikipedia, you'll get this very simple explanation: a non-metal with atomic number 34, discovered by John Jacob Berzelius in 1817. From an animal nutrition point of view, it's an essential trace element, which deficiency has been associated with things such as impaired growth, impaired fertility, and possibly poor health within farm animal species. And this health and the physiological effects of selenium are mediated by these things called selenoproteins. So a selenoprotein shouldn't really be confused with a selenium-containing protein. So we have selenomethionine. So selenomethionine can be incorporated non-specifically into body tissues, into body fluids, and that is a selenium-containing protein. A selenoprotein is a protein that is functional. It contains a selenocysteine functional core, so it's the catalytic site of the enzyme. It's not free roaming. It's not something that's actually found floating around in the body. It actually requires a genetic sequence for incorporation into a selenoprotein, so a selenocysteine insertion sequence. So the selenoproteins, 25 have been identified to date. The most well known I've put up here on the screen. So we have the glutathione peroxidases. So these are some of the most important cellular antioxidants. Thyroidoxin reductases. So they're involved in the reduction of thyroredoxin, which is a cell redox signaling mechanism. The deodonases, they serve a role in thyroid function, the conversion of T4 to T3, so they can affect metabolism. And then there is selenoprotein P. Selenoprotein P is involved in selenium transport. Approximately just over 50% of endogenous selenium is bound or linked to selenoprotein P. It's not only involved in selenium transport, but it has actually shown that it also serves a role in phospholipid or reducing phospholipid hydroperoxides. So what is the purpose of selenium supplementation? Well, primarily it's actually to support animal health. So many people, when they actually look at their animals, are not necessarily aware that they are selenium deplete or they are actually requiring some form of supplementation. And that goes for probably most trace elements. So this is a slide taken from Larson, or an image taken from Larson. And very typically, when you have an adequate supply of a trace element, there are no problems. And then you pass to a phase which is subclinical. Subclinical because there are no clinical manifestations of it. If you were to take a blood sample or you look more intensively at the animal, you might notice that there is a deficiency. So this is where you might expect to see, in the terms of selenium, changes in selenoprotein expression. As that selenium status or trace element status continues to decline, then you see the visual manifestations of deficiency. And with selenium, when you see those deficiency symptoms, you are probably quite chronic. And we'll talk a little bit about what types of symptoms you might see with fairly chronic deficiency. So in terms of the symptoms, so in very severe deficiencies, the clinical symptoms that might manifest themselves are things such as muscular dystrophy, cardiac myopathies, liver necrosis, and at very extreme levels, you might expect to see impairment of thyroid function. At this moderate, mild deficiency, so we're talking about the subclinical symptoms, there are no, as I say, no symptoms that have manifested themselves. But you find that if you actually look at some biochemistry, hematology, is that there is impaired antioxidant status, as you find there is downregulation of the expression of glutathione peroxidases or the thyroreductin reductases. You also might find that there is some reduction or some retarded growth, some impaired fertility, and that could be attributable to downregulation of selenoproteins P, N, W, and glutathione peroxidase 4. So selenoprotein P, I've already mentioned, is involved in selenium transport. Selenoprotein N is involved in protein folding, relieving endoplasmic reticulum stress. Selenoprotein W deficiency has been linked to muscle degeneration, and glutathione peroxidase 4 is quite important, as it's very important in maintaining cell membrane integrity. It protects the phospholipids. Carrying on with that, reduced or impaired immune or inflammatory responses, and they've been attributed to downregulation of selenoproteins K and S. Selenoprotein K is involved in calcium flux in immune cells. It's also been shown to have a role in T cell proliferation. And selenoprotein S, again, like selenoprotein N, has been seen to have a role in protecting the endoplasmic reticulum from stress, so again affecting protein folding. So although those things are things that may not necessarily manifest itself, it might be something that's affecting an entire herd, and because there are no clinical symptoms associated with it, it might be accepted or might be accepted as being the norm. So the thing about selenium deficiency is the symptoms that you see are somewhat reflective of selenium status and selenoprotein hierarchy. So a little bit more information about selenium hierarchy. So 3 important points. Firstly, not all selenoproteins are expressed to the same degree. So the first thing you have is what's called tissue-specific selenium hierarchy. So there are some tissues in the body that hold on to selenium for dear life. And those tissues are the brain and the testes. So the example I've given here, the figure at the bottom of the slide, shows the effect of actually selenium depletion. Admittedly, it was poultry, but poultry brains are easier to get hold of than cow brains. But what you find is that in animals where you deplete selenium, yes, there's an impact in terms of what you see in muscle tissue, but it's conserved in the brain. And similarly, you find the same in testicular tissue, but the same does not apply to ovarian tissue. There is no reserve. Selenium is not conserved in ovarian tissue, but a reduction in selenium within ovarian tissue does have an impact upon steroidogenesis. So secondly, there is the hierarchy of selenoprotein expression. So not all selenoproteins, as I said, are expressed to the same degree. They're all very much dependent on the selenium status of the animal. So as previously mentioned, the deodonases, they're fairly bulletproof. You need to have really fairly low circulating concentrations of those to have any really serious impact on deodanase activity. And then as selenium status improves, then what you find are the glutathione peroxidases. And there is a hierarchy within the glutathione peroxidases. So the one that is more resistant or more tolerant to low selenium would be glutathione peroxidase 4, and at the other end of the scale would be glutathione peroxidase 1. So as selenium status of the animal improves, you find there is better expression of those selenoproteins as status increases. Thyroreductin reductases, they sit within a niche or within an area similar to expression of the glutathione peroxidases when you're talking about selenium status. Then we come to selenoprotein P, and selenoprotein P actually requires a much higher level or a better selenium status to have maximum expression of that particular selenoprotein. And certainly in humans, the recommendation I think in humans is an intake of 65 milligrams per day, or micrograms per day, sorry, that's fairly fatal, but micrograms per day, where it's been shown that to actually get a maximum expression of selenoprotein P, you're looking at an intake of about 100 micrograms per day. So there's quite a marked difference in how that is expressed. And finally, where selenoprotein expression was often thought to be just a function of selenium status. So if you had adequate selenium, you had adequate expression. Well, actually, the oxidative stress or stress on the animal impacts how selenoproteins are expressed. So a couple of studies listed here. So polyunsaturated fatty acids and inflammatory cytokines added to cell culture were shown to upregulate the mRNA for GSHPX4, and the addition of peroxides to, again, tissue culture have been shown to increase mRNA for GPX1 and GSHPX4. So an indicator of oxidative stress upregulates mRNA expression. So this upregulation or expression of GSHPX mRNA is affected by oxidative stress, and that is independent of selenium status, whereas the actual expression of the selenoprotein itself is dependent upon selenium status. So the example in the graph shown below, soon to be published, this is looking at peroxidizability index. So we looked at the fatty acid profiles of muscle tissue, and from that we calculated the peroxidizability index of the tissue, and we looked at glutathione peroxidase activity. And what we found is there's a positive correlation between glutathione peroxidase activity and increasing peroxidizability. And that happens in animals that are selenium deplete. But in selenium deplete animals is that you have a similar peroxidizability index, but you have no response. So a very good analogy is that the mRNA is, if you like, an effect of turning on a beer tap and wanting beer to come out. But unless there's beer in the barrel, then nothing will come out. So mRNA is the tap, selenium is the beer. So if we come back and we look at reasons why we might cull dairy cows. So Phil's already touched on a lot of this, so I may be repeating things already said or touching back on them. But primary reasons for culling dairy cows is a failure to conceive, mastitis, and poor production. There was a study conducted by Hanks and Kusabayati. They were working within the University of Reading, and they did a survey of 500 UK herds. And within 75% of those surveyed herds, the culling rate was 24%, and exit was 3.7 lactations, which is, you know, it's a fairly short productive lifespan. So when you actually look at this failure to conceive, mastitis, and poor production, is that they are symptomatic of this thing called the dysfunction inflammatory response, typically during the transition period. And this has been attributed to oxidative stress. There's also been reported that this inability of the animal to actually adapt during the transition period results in systemic inflammation, immunosuppression. So oxidative stress, as I've already mentioned, upregulates selenoprotein mRNA, that's selenium independent, but selenoprotein expression, the proteins required to deal with oxidative stress, is selenium dependent. And what you are seeing is that in this subclinical phase is where these selenoproteins are not necessarily being expressed to deal with the problems or the physiology that the animal is encountering. So I apologize for how this slide is laid out. It did have animation, but I took it away because it became too confusing. But very simply, if we take oxidative stress, it's been linked to things such as retained fetal membranes, and that is a typical indicator of selenium deficiency. It's also been linked to elevated somatic cell counts, mastitis, and metritis. And certainly elevated somatic cell counts, mastitis, and metritis, they are linked in turn to reduce immune function. And reduced immune function has been shown to be dependent to some extent on the selenoprotein expression, or the expression of certain selenoproteins, particularly those that are involved in protein folding, cell signaling, and calcium regulation. So one question we might want to ask, is the selenium supply that we give our animals, is that actually adequate to meet the selenoprotein expression requirements of modern productive genotypes? And the reason why I say this is that at the bottom of the screen, there are two graphs. These are admittedly based on poultry, but it tells a very interesting story, is that according to the NRC, these two species of poultry have identical selenium requirements. But when you actually look at how they deal with selenium, they actually deal with it in markedly different ways, in that one actually manages to accumulate it, one doesn't necessarily accumulate it, doesn't deal with it particularly well, which suggests that as we have moved on with our dairy cows, most recommendations are quite dated. So are they fit for modern genotypes? So that the NRC recommendation says it's somewhere approximately 0.3 milligrams per kilogram. The legal US limit is 0.3 milligrams per kilogram. So you can't actually feed any more than that anyway. But glutathione peroxidase responses have actually been reported up to 0.4 milligrams per kilogram. Now that tends to suggest that the recommendations, the limitations, do not actually reflect the needs or the physiology of modern high-yielding dairy genotypes. So what about how do animals respond when you give them selenium supplements? So the response that you see will be dependent upon the selenium status of your animal prior to supplementing it. Is it actually showing clinical symptoms? Therefore, you might see a marked improvement. Are the symptoms subclinical, whereby actually what you need to look at is some marker of selenium status. And the marker that you choose is going to be affected by the selenium status of that animal and what remedial action you may have taken. Are you measuring in blood? Are you measuring in milk? Are you measuring in plasma? Are you measuring total selenium, or are you measuring a selenoprotein or another biochemical indicator? So they will have an impact on the decision that you make. The status of the animal itself is going to be largely influenced by your geographical location or the selenium soil content. So in the US, I understand there are areas where there are fairly high selenium contents. Equally, there are areas where selenium content's quite low. Here in Europe, which we were once part of, I believe, selenium contents in the soil are fairly poor. We're known as old soil types. So selenium status in the UK is generally poor anyway. So the management system that you have is going to affect the selenium status of your animals, particularly the geographical location. So how much access do your animals have to graze pasture? So if your animals are quite extensive and they're grazing quite large amounts of pasture, is that if your soil selenium is fairly low, then their selenium intake is also going to be fairly poor. In terms of the diet types that you're feeding, are you feeding a forage-based diet or are you feeding a concentrate-based diet? If you're feeding a forage-based diet, that's going to be locally conserved forages, most likely, and that will reflect soil selenium conditions. If you're feeding more concentrates, are those concentrates commercially sourced? They may already contain a selenium supplement, or are they made on farm and are you adding a selenium supplement? Are you feeding your diets as a total mixed ration, or are you feeding them as a forage on the floor and then top dressing your concentrates? Because the difference between the two is that a TMR for a comparable dose of an identical source, there can be 30% difference between the responses that you see between feeding it top dressed and feeding it as a TMR. So how you feed it can have quite a profound effect. Then we come to this question about supplement use, the supplement source. So principally, selenium supplements come in two primary forms, shall we say. So there are the ones that are termed inorganic, and those are typically selenium salts, sodium selenate, sodium selenite. And then we have the organic, and these are typically the ones I'm talking about, selenomethionine based. Now, in terms of how effective those sources are, it's not really the topic here, but generally the responses to organic forms, selenomethionine based forms, give a twofold difference in terms of responses, and they actually occur in a much shorter period of time. And the organic sources tend to be considerably less toxic than the selenium salts. So in terms of the fate of ingested selenium, so this is fairly simplistic. I borrowed this from Margaret Raymond. But very simply, it doesn't matter what the source of selenium is, whether it's organic or whether it's inorganic, is that regardless, is that all selenium to be used as a functional selenoprotein needs to pass through the selenide pathway. For inorganic sources, when passing through the selenide pathway, they either use for incorporation into selenoproteins. If they're not used for the incorporation into selenoproteins, then they are methylated and excreted from the body. So they're either used or they are lost. They're not retained. In terms of the organic forms, so these are those that are predominantly selenomethionine, is that yes, selenomethionine still has to pass through that selenide pathway in order to be used. But one benefit of selenomethionine is that it is non-specifically incorporated into body proteins, body tissues, and it acts as an endogenous selenium source during times of deficit. So as the animal undergoes natural protein turnover, that releases selenium back into the systemic circulation and provides a buffer, if you like, for animals that might be extensively grazing. So this is a rough and ready graph. This is unpublished data, but this is a collection of data which I think worked out to be about just over 300 dairy cows. And this is the post-supplementation period. So the dotted red line at the bottom represents animals that were unsupplemented throughout the period of the study. So generally poor. And this hatched red line that sits just above it is the level of selenium that is required to actually have reasonable GSHPX1 expression. So when we look at those animals that have received inorganic salts or selenium salts, yes, it's elevated over control animals. There is a decline. I know I said you use it or you lose it, but what you have in there is that within the diet, there's background selenium, and that's organic in nature. So there's a little bit of inorganic and organic tied up in this particular line. In this green line, this is organic selenium, selenomethionine. And so if we actually look at glutathione peroxidase, and admittedly this is an extrapolation, is that if we look at how long post-supplementation that glutathione peroxidase or whole glutathione peroxidase levels are maintained to a point where they might become of concern, is 100 days. Whereas when you feed selenomethionine-based supplements, what you find is that's extended by a further 75 days. So it gives you a total of approximately 175 days coverage when using or when comparing it to no supplement at all. So those are probably the benefits for the animal on the ground, certainly protecting her. But then there is this question of, well, what about the implications that the dam selenium status has on the calf? So it's not unknown, it's been fairly well published that there is a significant correlation between the selenium status of the dam and the selenium status of the calf. And there are a number of studies that have shown that with improved dam selenium status, you have improved calf selenium status, certainly in terms of total selenium in whole blood and plasma. In a number of studies, that's translated into better or higher glutathione peroxidase activity, so that shows better selenoprotein expression. And in some studies, they said that calves that have received, or calves born to dams that have received selenium during the latter part of pregnancy have increased vigor scores. But more recently, so this is a study conducted by myself and others that looked at hydroxyselenomethionine, which is a selenomethionine analog. And what we found was, yes, it increased the selenium status of the dam and of the calf. Glutathione peroxidase levels were enhanced. But we stopped and we actually looked at the selenium content of colostrum. And what we actually found was that with animals that were fed selenomethionine from dams that have been fed hydroxyselenomethionine, is that we had elevated colostrum selenium. Well, again, that's not anything new. There are a number of studies that have said that this selenium content is higher in first milk than it is in following. And when you might ask the question, well, what is the benefit of that? Again, there are a number of studies that tend to show that actually intestinal transfer of IgGs or of albumin tend to be improved when the selenium content of the milk is actually elevated or higher. And this is, well, confusingly, whether the selenium is enhanced naturally via the dam, or whether you actually add selenium to the colostrum itself and then feed it to calves. What was of particular interest is that generally when you feed selenomethionine to dairy cows, is the predominant selenized amino acid that you find in the milk is selenomethionine. But when we did speciation on the first milk, what we actually found was that the predominant selenized amino acid was selenocysteine, not selenomethionine. And if you recall from my very first opening statement, is that selenocysteine is indicative of a functional selenoprotein. So digging around, trying to understand a bit more about this, and admittedly, this was not in cattle, but this was found, a study done in mice, is it was reported that selenoprotein P, so that is a selenoprotein, is the predominant selenium transport protein in murine milk. And it was postulated that it was actually an important mechanism for the maternal transfer of selenium to the neonate in that early postpartum period. And if you think about it, is that the consumption of immunoglobulins in that very narrow window postpartum, that's through pinocytosis across the gut wall. Well, the selenoprotein P is a fairly large molecule, and it might be that also passes the gut wall through pinocytosis, and that must confer some benefit onto the calf in terms of selenium status. I suppose, sort of moving on, and this heralds again back to a little bit of what Phil had to say, is that there have been a number of studies conducted in a number of farm animal species, and the ones that I'm most familiar with are those that were conducted in sheep or in pigs, that have shown that any nutritional insult in that first trimester of pregnancy, so an initial nutritional insult of the dam, has an impact on the offspring. So one, it has an impact on placental development. So poor nutrition can actually enhance placental development, ironically. But that is driven by the fetal side of the placenta. And it's also shown that when you actually look at the offspring postpartum, is that in that first generation, there is an alteration to energy nutrient partitioning. In fact, what you find is that the neonate actually finds it very difficult to deal with things such as glucose. So that was admittedly in sheep and pigs, but more recently, Opsheimer, I listened to a talk by him a year or so ago, and they're now looking at this in dairy cattle, and they've shown actually that these pre and postnatal insults can actually have long-term consequences for the growth, fertility, milk yield, and longevity of dairy cows. So the dairy cows that are following on as the next generation. And these are reflecting very much the observations that were made in sheep and pigs 10 or 20 years ago. So what I offer up now is maybe another thought, which is intergenerational effects. So where we mentioned that we have this impact with fetal programming, so that you apply a nutritional deprivation to a pregnant dam. The F1 generation shows altered energy metabolism. But if you take the F1 generation and you breed from them, you don't need to induce any form of intervention, but the F2 generation has the same altered energy metabolism. So what you have here is where you have a macronutrient that has affected not just the first generation, but it affects the subsequent generation. And if we think about how we breed replacements for dairy herds, you do have grandparents, daughters, and the population moves on. So if that's the impact of a macronutrient, we're only just discovering impacts that micronutrient deficiencies are actually having on animal performance, animal physiology. So is it likely that sort of these small deficiencies are having an impact on animals in the longer term in multi-generation steps? So I'm going to draw a close to it. Our time is moving on. So I thank you for your attention, and I move back to Brian.
Thank you so much, Darren, for that excellent presentation. So now we will open the floor. Everybody will be glad to know I have at least 11 questions, so nobody's credits are at risk here. So but keep the questions coming in, particularly for Darren, because I see that We have quite a few for Phil, but keep them coming in for both speakers, but in particular Darren, if possible. So let me go to the Q&A. So a first question for Phil is why MUNs only affect first AI, but not subsequent AI in the study that you show?
Sure. I think that's as cows move through lactation, the milk components, they change. And I think as well, the concentration of that nitrogen circulating, or pretty much cows are going to get into peak. So around that first breeding, that's where we expect that cows are going to be in their highest milk, or just after that. And as times goes by, that milk yield probably goes down, right? As lactation curve goes down. And then maybe that's not as representative to change that uterine environment, for example. So I think one of the things is associated with that, and perhaps liver status starts getting better, so you have more hepatocytes free to deal with the nitrogen clearance, and uterine tissue as well. So I think it's more related to that, I think. But they don't have any conclusion, any hard conclusion, because it was just a epidemiological type of data, not so much doing the controlled research.
Brian, you're muted.
Yeah, big mistake. I always do it once at least, so bear with me. One for Darren. What would be a good or normal level amount of selenium, and what would be considered too low or too little?
So a very good question. So what would you consider to be too little or too low? So we've mentioned that for productive dairy cattle is that selenium response has been shown to be 0.4 milligrams per kilogram of dry matter, which is above the US recommendation. I would suggest that maybe what's too low, again, what is the purpose of the animal? How metabolically active are they? So if you're talking in terms of high yielding animals, I would probably say that you need to feed whatever the maximum recommendation is that you can feed to meet those requirements of those animals.
Thanks, Darren. Phil, another one for you. You talked about PUFAs. According to your observations, or at least your reading of the literature, do you think PUFAs are essential for early embryonic development?
Yeah, that's a very good question that I think we definitely have a lot of information out there. I think there is an overall recommendation to include some PUFAs to diets of cows. We're not sure 1 pound. There's a lot of anecdotal information about it, but point being is that some of those polyunsaturated fatty acids, especially omega-3s, right? So we can design products that have these specific types of fatty acids, and we can, or we have to protect those, because if not, remember, in the rumen, everything is going to get saturated, so you're not going to end up with anything being absorbed by the cow. But there are some research saying that, you know, if you're feeding more of those PUFAs, you can have some of that prostaglandin production being prevented, and that would lead the tissue to convert more into cholesterol, and that could lead to more progesterone. So just by itself could lead to a better embryo survival. So there are classic research in Florida, but I didn't see that being that have been repeated again. But I think that's kind of where it is. If there is that chance of that lipid profile to be more unsaturated in the uterus, that could lead to a better embryo survival based on this theory of cholesterol and progesterone. If you remember right, I showed you data that the cows that consumed methionine, they had a higher expression of that LCAT enzyme that is the one converting cholesterol. So that would kind of go along with this theory that I'm just talking to you. But as far as so far it's hypothesis, but there are a lot of products out there playing with that amino acid, fatty acid profile and prevention of prostaglandin production.
So yeah, I think you already really answered the second question that's here, but I'll just ask it anyway. You can add to it if you want. So is there any interest to, do you think, to feed methionine or more methionine with specific fatty acid supplements to get, let's say, the best effect?
That's a good point. I'm not sure. I want to assume that if we are feeding amino acid and that profile is changing, it's because it's needed. I don't think it would change just by chance, right? Or just to store. That's not the type of fatty acid would be stored. So I think some of the signaling that's needed. But we are always comparing supplementation with no supplementation. So, yeah, I don't know.
We'll leave you to think about that. Okay, Phil. Okay.
Okay.
So, another one for Darren. Do you know if selenomethionine and methionine would affect other animals' health and reproduction similarly? For example, horses instead of dairy cattle. You're on mute, Darren.
I was hoping you could lip read, but there we go. Going back to your question, is that horses is a good example to choose. The problem with horses, they're actually quite sensitive to selenium. In fact, I think anecdotal evidence says that Custer's last stand was because of selenosis in pack horses. There is evidence to show that actually selenomethionine in horses can have a beneficial effect, if we're going to use horses as the example, is that the study which I mentioned where we looked at 300 dairy cattle and removed the supplement and looked at post-supplementation, the effects on glutathione peroxidase activity, is you get the same effect in horses. So you do have that protective effect. So I would say, yes, you would see it in other animals.
Okay, thank you, Darren. Phil, do you have any evidence whether methionine supplementation could be beneficial to rule out the negative impact of heat stress on fertility?
Very good point. And I'm trying to think about it here, and I don't have that answer straight. I have some things that may connect to that and help, but I think we do need that data a little bit more specific. So we know that if we have heat stress, and that was done in Canada with metabolic chambers, they induce heat stress, and they saw that plasmourea nitrogen goes up, and that the profile of amino acids, including methionine, goes down. So cows in heat stress, they seem to have a lower concentration of some amino acids, including methionine, in the circulation. We have done one trial here with the heat blankets where you can induce heat stress, and we saw some positive effects when cows are under heat stress. They are par-fed, so that means we take out all the intake confounding, and it seems like when they have the rumen-protected methionine in their diets, when we did mammary gland biopsies, we see that the alveoli, when you see which ones are proliferating, which ones are dying, that number and the association, if they get methionine, they have more more proportion of those alveoli that are functioning. So I think there is some effects on, for sure, on the mammary gland, but I don't have any idea specific to the uterus or to the follicular dynamics. But I would guess that, yes, there's something there.
Thanks, Phil. Perhaps it's another one on for you. On the embryonic losses and the embryonic size data you shared, what is the mode of action of methionine, and why is there no effect in primiparas? Okay.
And I think one logical one is for us to think just like hypocalcemia, why hypocalcemia happens more in multiparous cows than primiparous. So I think it's just a intake and output issue. So multiparous cows, they're going to be producing more milk. So you have more methionine being excreted. So I think that's where you're going to see more of that methionine being needed for the milk protein and not so much getting into the uterus. And if you really look to that number, when you compare multiparous with primiparous, the number of the volume is pretty similar. it seems like the control multipairs that was lower. That means that they were suffering and not getting enough of that amino acid. So I think it's just milk yield that is justifying how much is being partitioning. I think that's it.
Thanks. Another one for Darren. Do you know what the relative safety / toxicity is of the different selenium sources, inorganic versus seleno yeast versus selenomethionine versus the hydroxy analog of selenomethionine? That's a real difficult question, I imagine.
It is a difficult question, Brian. So very simply is that for inorganic sources, there's quite a lot of information about the LD50. And typically LD50s are measured in lab animals such as rats, mice, rabbits. In terms of the LD50 for inorganic sources, ranges between 5 and 8 milligrams per kilogram of body weight. For selenomethionine, data is a little harder to come by, but generally speaking is that the organic sources, are actually less toxic. So I know something about the seleno use, is that in terms of, I think, European, is that I think the inorganic forms are category 2 toxins, whereas the seleno use were classified as, I think, category 5, so considerably safer. So in fact, it is this ability of the body to actually store selenomethionine that actually makes it less toxic, because the body doesn't need to methylate it and get rid of it.
Thanks, Darren. I think we'll take do another couple if that's okay with the audience and with Christy. How much methionine should be given in feed to protect from bad inflammation?
So I think that what we've seen now is, and it's kind of interesting to see the papers and everything that was published and how we design and formulate diets maybe 10 years ago and how we do it today. It's a little bit different, right? But I think if we keep to the recommendations that I've mentioned about that association with methionine to energy that we are feeding, I think the models, they predict energy pretty well. I'm not sure about protein requirements and amino acids yet, but I'm pretty sure that if we tie that grams of methionine to the energy that the model is calculating, then we're going to be doing a better job than we did before on percentage of MP, or before on just the ratio of lysine to methionine. So I think we've been growing on how to do that. But I think this bad inflammation, I think the cow can take care of it if she has the pieces like amino acids to handle and produce the antibodies and all the cytokines and cell proliferation that she needs to do if we feed at this point. But who knows, 10 years from now, I may be saying something different. But for now, that's how we are doing. That's how the results you're seeing, how we are getting.
Phil, I'm sure you'll be saying something different. So maybe a last one for Darren. Can inorganic selenium satisfy the selenium status and permit sufficient de novo synthesis of selenoprotein, like some data suggest? Oh, no, sorry. Some data suggest that this may be up to 40% of selenoproteins end up in milk.
So the short answer is
clear.
Yeah, it's fine. So the short answer is that yes, inorganic selenium can satisfy selenium status. The point that was trying to put over is that inorganic selenium sources are actually quite short-lived. They're either used or they're lost. The benefits that you have with selenomethionine-based ones is, one, you have this endogenous supply, which you do not have with inorganic forms. Secondly, there is this issue of toxicity, is that selenomethionine is less toxic than selenium salts. So although you can say that, yes, you get a similar response in terms of de novo synthesis of selenoproteins, is that everything goes down that selenide pathway, is that in terms of the longer term protection and probably response efficacy, that's where you see differences between the sources. So responses that you see in organic forms is much more marked, I would say much more rapid than you see from inorganic sources.
Okay, and one last one to finish the webinar. Phil, the levels of methionine and lysine you presented relative to ME or as a ratio, have they been developed solely to optimize milk response? And if they have, will the recommendations, are the recommendations different to optimize reproduction?
I think that's a very, very good point. And I'm not sure if that was or, but that's grams to ME, not so much the ratio. We still have the ratio, but I would focus on the grams to the ME, right? And then you adjust your lysine based on that ratio and make sure you are within that grams to ME. And that's a very good point because as I showed that little graph there that talks about the efficiency, how much I feed, how much the cow is going to be using, and that's the most efficient that translates to how much I should feed. However, if you guys follow Formula One or any race, car race, we're not talking about efficiency. We don't want to be efficient. Those guys are going to the limit of performance, right? So sometimes you may not want to be on that most efficient. You may want to go to the better performance. And I think that's where any model fails a little bit to tell us how does that happen. We are requiring the minimum, what the cow needs to perform or to survive, right? So to be honest, I don't have your question. Right now, we are using data from lactating cows to feed dry cows. That's what we have now on that ratio. Perhaps it should be more, perhaps it should be a little bit less. What you're seeing is this huge difference between control and what we feed, and it seems to be doing very well, right? But to optimize performance, maybe we should be feeding a little bit more or less, but I don't have that answer yet. But I think for sure, we need to get everybody to the same level where we are feeding that needed methionine. That's not a full reality yet, in my perspective. So we need to get comfortable there, and then we can start talking about high performance, low performance, reproduction, and try to see what is that difference there. With everybody being fed methionine, see who are the highest performance, lower performance, and we have some more epidemiological data to get started maybe. That's a good suggestion, good point.
Well, thank you so much, Phil and Darren. On behalf of the audience, I'd like to thank you both for the excellent webinars you gave and the excellent answers you gave to the questions. Just to point out that if the question was not answered, they will be answered subsequently. And so I'll just explain what the next step is in terms of providing additional information. There has been a recording of this webinar, which will be made available before or on October 24th on the DCRC website. In addition, it will also be made available on Adicio's feed channel online, where it will be posted with a document that contains all of today's questions and answers. So without, unless Christy has anything to add, but I just want to remind you all that of the upcoming annual general meeting of DCRC on the 9th to the 11th of November. It's a must see, a must do. So for all of you that are members, please, if you have not signed up, please sign up in the very near future. So again, thank you all for participating. And if there's any questions specific that you want to pose to Adiceo, please feel free to contact Christy and she will give you my contact information. So thank you again, and good morning, good night, and good afternoon. Bye.